Yeah, great. Yeah, so today I wanted to give an update in terms of a non-human primate working group that's been formed from the SCORCH consortium, leveraging the 4 projects that are generating single-cell data sets from the non-human primate model. Um, Today we'll break it into 2 parts, and I'll share the presentation with our colleagues at UNMC. I'll focus mainly on the first part, just overviewing particularly the non-human primate data sets that are being generated for SCORCH. And it'll kind of give you some insights in terms of having the ability to look at one particular region that seems to be harmonized across all the data generation sites from non-human primates, focusing on one virus, SIV, for non-human primate infection. And then in part 2, the group has really been, I think, asking some questions in terms of the challenge of finding infected cells, in terms of looking at a particular region across multiple groups. And clearly, the more cells we go through, the more animals or even people that are being surveyed, the better chance of finding infected cells and potentially looking at cell signatures related to infection. And then I'll hand over the presentation, and we'll discuss a little bit about the feasibility of processing cryopreserved nuclei for the multi-omic assay across multiple sites, in terms of just kind of being able to utilize nuclei for other assays or scaling up additional cells that can be profiled. And then Meng will also talk about evaluating ambient RNA removal tools for some of the data that has been generated for one of the projects. So this slide here kind of shows you the survey of the non-human primate studies that are being pursued for data generation across 4 different UO1s, including a supplement to the Cornell site. At the top there, you can see the teams that are leading the projects. We have a team at UNMC, a team at Weill Cornell Medicine, collaborations as well with Weill Cornell and Johns Hopkins University, and then also a collaboration between UCSD and UNMC. All the groups are utilizing a rhesus macaque model. The virus being utilized in these different model systems are utilizing SIV MAC251 across all the groups. You can see there also, in terms of the drug exposure, it's nicely been organized in terms of being able to compare and contrast across different drugs being administered to the animals in conjunction or alone as control conditions. When you consider, if you start to look at the potential interactions between the drugs of abuse and infection, you have conditions where you can mix and match morphine exposure with SIV or morphine exposure only. And additionally, looking at antiretroviral treatment as well, animals that are suppressed. We have a group doing morphine exposure at UNMC. The Weill Cornell Medicine group is working with the group of affirms utilizing oxycodone in that particular model. There's a supplement that's been added to generate additional data from the hippocampus, which is morphine exposure, which we'll tell you a little bit more about today in terms of utilizing some of that data. There's a new project that just got started utilizing a model that's administering oral THC or cannabidiol to non-human primates and looking at the hippocampus as well in that project. And then a newly funded award as well at UCSD and UNMC, examining cocaine. When we look at the brain regions that have been focused, as you could imagine, the assays to generate the data are quite costly. So, it was, I think, a strategic plan to try to harmonize as best we could across all the groups, a particular region of interest to start to really get data that would be able to be integrated together in some, I think, publicly available analyses, but also too as individual projects piece things together for validation to see which potential drugs may be doing things that are similar or dissimilar to single cell states. The one that you can see highlighted there is the hippocampus, which for most, in the neuroscience field, is one of the regions that neurogenesis does occur. And it gives us, I think, another unique opportunity, which I'll talk about in a subsequent slide, to build on cell annotations generated from a Yale SCORCH team member in terms of atlasing the hippocampus across species, which has been, I think, widely available publicly as a data set that really can be integrated with what SCORCH is generating here. And then you can see at the bottom, the major technology that the groups have chosen to use has been the multi-omic assay, so paired RNA and ATAC. As I said, one of the unique things, as you look at that slide and see across the groups the data that will be generated and is currently being generated, it really fits nicely with the Yale group, Ninad's atlasing across different species of the hippocampus. So, this data set here really can be integrated in with what SCORCH is generating, and really, I think give us kind of an additional aspect to moving beyond the atlasine that's been occurring and looking at infected cells and drugs of exposure. Excuse me. In terms of the data set we've been able to look at currently, we've utilized the data set where we looked at morphine-treated animals that were infected, as well as animals that were either vitally suppressed or untreated. And as you can see here, one of the things we've observed in the working group has kind of been the difficult challenge and stochastic nature of detecting infection in the CNS. Particularly here, I'm demonstrating data from the hippocampus. Depending on kind of where you look, I think it's a needle in a haystack issue, which a lot of groups have highlighted. But in this context here, I've highlighted 3 different animals which we have been able to detect positive SIV infected cells in microglia. And as you can see, in the groups that are not left untreated, only 1 animal was positive for infected cells in terms of surveying over 50,000 nuclei across 4 different animals at 50,000 or 40,000 reads per cell. And it really highlights, the complex interplay between opioids of abuse and SIV infection in the NHP model. And this was a nice review by one of our consortium members and a member also of the NHP group, reporting, I think in the literature, the controversy between morphine infection and the number of infected cells that are detected in the non-human primate model. And what we have observed in the multi-omic data is when there is virus, it's in microglia. And the 10x genomics multi-omic assay detects both SIV RNA and TN5 accessible SIV DNA. Here I'm showing you from that 1 animal what we're able to observe when you do see infection. On the left-hand side there is just the microglia cells that were captured in the multi-omic data set from the hippocampus. Those cells are highlighted there are the ones that are detecting SIV RNA. And on the right side here, it's an IgV view of the virus SIV. So it's around 9,000 base pairs. And the mapping, the histogram is the RNA reads that are mapping to the virus. We do get good coverage of SIV at the RNA side, as well as at the DNA side. So those cells that are actively transcribing SIV RNA in the hippocampus and microglia are accessible. So the TN5 inserts in SIV, and we're able to capture that on the multi-omic assay. So in terms of the working group goals for the NHP group, I really feel it's to chart the discovery or validation of signatures of the distinct or synergistic effects of drugs with SIV and potentially in the HIV data set that the human groups are working on. And it's to harmonize approaches and regions of interest generated across the NHP groups, as I demonstrated a little bit of our effort to do that already with the hippocampus as a focused region to start with data generation. So I'll hand over to Meng and let him talk about the feasibility of sharing cryopreserved nuclei across multiple sites.
Okay. Okay. Is it showing the correct one?
We see the other one if you just want to swap it.
Okay. There we go.
Looks great.
All right. So the sample is prepared at Cornell, and it was sequenced there, and then it was sent to UMC for another set of sequencing. So here we did some preliminary analysis for comparison. We used the cell ranger pipeline with the combined reference genome of rhesus monkey version 10 and the SIV MAC 251 from the RefSeq. And we pick up here. I picked a couple of parameters to show that in this table that even with the variation of the sequencing depths, the quality of the cells are very high-end, consistent. See that the mapping rates, called peaks, etc. Notably, the fraction of the transcriptomics reads in the cell from both sides is on the higher end. And you see the figure at lower place, the joint cell calling, which is based on the RNA UMI count versus the ATAC transposition event in peaks per barcode. They're comparable for both of the samples. Then we aggregated the same sample from 2 different sites. You can clearly see that from both ATAC and the gene expression plot, the major cluster groups of cells from both sites are comparable even without any batch correction. This is just preliminary analysis. But the high quality and the consistency shows a promising future use of this technique, which can be applied to different tissues, species, and the other techniques to explore our samples. So when we were dealing with our single nuclear RNA-seq data, we were having some issues with the embedded RNA, and here's some process that we've been how we deal with this problem. So this is some background I borrowed from the figure. I borrowed a figure from the Cell Bender paper, which is published on the Nature method. So when we prepared a single cell or single nuclei for sequencing, the RNA molecules from the ruptured or degraded cells are captured by cell-free droplets, also those cell-containing droplets, and form source of background noises. And that messes up 2 things. One is the cell calling process, and the other is the gene expression pattern in the captured cell. And these RNAs inside and outside the droplets are referred as ambient RNA or floating RNAs. In the experiment of the mixed species mouse and human, we can see that in the blowed up, the hundreds of off-target human transcripts showed up in the mouse cells. And it's an even bigger problem for single nuclei RNA-seq, for the harsh isolation protocols that produce ruptured nuclei and a high concentration of cytoplasmic RNA in the suspension, where you can see in figure D, the lower UMI count where the empty droplet is supposed to be, but their exon fraction rise by counting the embedded RNAs. If we take this as it is into the downstream analysis, we will get inaccurate cell calling, wrong fold change, difficult for cell typing. Of course, if you see, you will find a lot of marker genes expressed everywhere. So when we are processing our single nuclear RNA-seq data, we found some of our data shows low fraction of reads in cell percentage on the left side, which indicate there are embedded floating RNAs in the samples. We also lost a nice first job in the barcode rank plot, which is supposed to separate the cell from the background according to the cell ranger pipeline. So we started to test some popular tools to figure out the embedded RNA. There are many methods available now for embedded RNA removal. So in the Cell Bender paper, it already compared with the DCON-X. So we start with the Cell Bender and the SUP-X. Cell Bender uses a machine learning model that is used to train the background RNA profile, distinguish cell containing droplets from the empty ones, and retrieve cleaner gene expression profile. Since the model spits out the probability of the droplet containing a cell calling process is also included, which I like a lot. SUP-X is a method for quantifying the extent of ambient RNA contamination while purifying the true cell-specific signal from the observed mixture of cellular and extraneous mRNAs. So for testing, we used the data from two samples of the middle frontal gyrus from the monkey and applied the same quality control criteria. Then used the software Partec to analyze the data. So you see here the graph-based clustering method gives a similar number of clusters from different methods. Now we want to take a look at a cluster based on the marker genes and what is the effect of the removing the embedded RNAs. So first we use the pan-neural markers, RBFOX3, SYT1, and the GRAN2B. So you see here the cluster 2 and 5 from the Cell Ranger unfiltered, cluster 2, 3, 4 from the Cell Bender, and the 2, 5, 6 from SUP-X stands out. Well, the cell with markers like these are that has very high expression level. If they are ruptured, the released RNA will get picked up by the rest of the droplets. And it is what we see here. So in the unfiltered one, all the rest of the clusters show certain level of expression of these markers. While after the cell binder or SUBX is getting cleaner. Well, cell binder perform a little bit better than the SUBX. Then we dig a little bit more to check if the neuron cluster can be separated into inhibitory and excitatory neurons. So as shown here, it's easier to tell the difference between the two after filtering out the ambient RNAs. Then we look at oligodendrocytes by marker MOG and the PLP1. It's the same observation that expression across all clusters even, they're not supposed to have it. But after filtering, the expression of the markers are more specific. Then we check the marker for microglia. The point here is that if the expression level of the marker gene is not very high, they will have a smaller proportion in all the free-floating RNA, and the effect of overflowing to other clusters will not be that obvious. So after this test, we decide to include cell binder to our single nuclear RNA-seq data analysis. Well, with that, I'd like to thanks to the members of the task force and all the labs from UMC, Cornell, Johns Hopkins, and that's the supporting funding. Thank you. I'm ready for questions.